You found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there YouTube, the Dapper Dinosaur here. Let's talk about the Institute for Creation Research again. They recently, at the time of my writing this, conveniently put their That's a Fact short videos into an hour and 40-minute long video compiling 62 such smaller videos. I figured we could look at about 10 per episode as a fun series where I'd get to cover a wide variety of topics. I've seen some of these, but I don't really remember them very well, so for the most part, I'll be watching them with you. I should note that the whole video uses music extensively and that I've had to do some pretty heavy auditing to get rid of most of it to avoid copyright problems. As a result, the audio is not very good. I hope that's okay. Our first clip is about human and chimpanzee DNA.
We've all heard the theory that chimps are close human relatives, but is there any truth to that?
We have, and for good reason. As some creationists like to point out, DNA is what's passed down sperm and egg to new generations. It's where the nuts and bolts of evolutionary biology happens. I'm not aware of anyone who disputes that DNA tests from places like 23andMe or Ancestry.com cannot identify close relatives. People have found siblings lost to adoption, they found cousins they never knew about, discovered ancestry from parts of the world they never expected, etc. It's an observable reality that genetic similarity indicates the degree of relationship between organisms. Creationists do not generally disagree with this either, and in the case of non-ape groups like the felines, they're happy to say that genetic similarity between, say, the tiger, Panthera tigris, and the lion, P. leo, indicates they are more closely related to each other than either is to, say, the bobcat, Lynx rufus. Indeed, the genetic difference between tigers and house cats, which are supposed by most creationists, including Answers in Genesis and the Institute for Creation Research, to be genetically related, is more than between humans and chimpanzees. It is in fact pretty close to the genetic similarity between a human and a gorilla. The evidence for human common ancestry with the other apes is stronger than the evidence for the common ancestry of most groups creationists themselves think share common ancestry. It's only the dogmatic assertion that humans as a species are a separate creation that comes from a selectively liberal interpretation of Genesis that makes creationists think this, and they're extremely inconsistent about it.
Secular scientists claimed in the 1970s that chimp genomes are 98% similar to humans.
Well, that was the headlines, but of course pop science reporting is terrible. But if you read the actual papers from then and now, that number is more specifically the percent similarities in alignable regions with insertions and deletions treated as single changes. I'll use a pair of books as an example. First, if you didn't know, the King James Version of the Bible is a revision of the earlier Bishop's Bible, and the two texts are nearly identical in most areas. But let's say that you took two Bibles, one a Bishop's Bible and the other a KJV, and decided to rebind them, but in a strange way. First, you mix in a bunch of pages with just random letters, punctuation, and spacing. Then in each book, moved a few sections around here and there, so they're no longer in the same order. Now, if you wanted to compare how similar those two books are to each other, on a per-letter basis, starting from the front of each, you'd get a very low number. But clearly, that can't be all there is. That doesn't capture the fact that many pages in one book are nearly identical to pages in the other, and that while there is some change in the ordering of such pages, they are still similar in their order. These nearly identical pages are alignable. We can look at both and show that they are so similar that in important ways, they're the same page. This holds true with the genomes of organisms. Interspersed between non-sequence constrained regions of the chimp and human genomes are constrained and alignable regions, where we can tell that, say, a set of genes in a human are really the same genes in a chimp, because they're in the same or similar order and have very similar sequences. We can't really align the bits of random noise, but also when we do align the parts we can't really say that the books or genomes are just a certain percent similar overall. So now that we have a better understanding of how and why the comparisons are done to come up with the percent similarities, we'll see what else ICR has for us.
And it was apparently verified by more modern techniques.
Yep.
But that estimate actually used cherry-picked segments of DNA that we already share with chimps, not the whole genomes.
It's not cherry-picked, it's the regions that you can compare in a meaningful way, just like with the two Bibles. You can't just compare the genomes in bulk because there's no obvious place to start or way to line them up for comparison. In fact, even creationist attempts to do such comparisons that have as a goal finding low percent similarities still focus on alignable regions. There really isn't another option.
The latest comparison to include all of the DNA between the two species revealed no more than an 85% identity.
This is a reference to work by Jeffrey Tompkins, a man I've already made one series about. In that, I called him the creationist who can't math, and for good reason. He routinely makes math errors ranging from a 3rd grade level to high school level. Did he do some kind of special whole genome comparison in getting this 85% number? Well, no, he did not. He also used alignable regions of the two genomes, because that's really the only way to do it in a way that gives comprehensible results. Further, he has run this comparison twice, each time making different but similarly boneheaded mistakes. The first time he used a bugged comparison program that treated insertions and deletions not as a single change, but as causing many, many changes. Let's return to our Bible example. Let's say that you're looking at both books' text of Daniel, one of the shorter books of the Bible. In one of the Bibles I own, it takes up 15 pages, so let's just go with that as the length of Daniel, even though it varies. Let's say that in the KJV plus random stuff book, we find that Daniel is set on 7 double-sided pages. But in the Bishop's Bible plus random stuff, the 3rd such page is missing, so in that Bible, the book is only 13 pages long. According to the bugged program that Tompkins was using, every page after that difference would be counted as completely different, and the similarity would be very low. Even more extreme, if you and I both had copies of the KJV, but mine was missing the N in the beginning from Genesis 1:1, Tompkins' program would return essentially a 0% similarity, even though the entire rest of the text is 100% identical. This was pointed out to Tompkins, so he decided to fix it and try again. But this time he didn't bother to weight his similarity results by sequence length. Continuing the analogy, this would be like finding that 2 Bibles had completely identical books of Jeremiah, Genesis, and the Psalms, the longest books in the Bible, but that their copies of Nahum, one of the shortest books in the Bible, were only 30% identical. Tompkins, in comparing these 4 books, would consider this a 92.5% similarity, but normal people looking at the lengths of the books would count this as a 99.4% similarity, based on the Hebrew word counts of all those books. Tompkins' method overestimates the difference by 93%, nearly double the difference. Let's actually dig into this a bit. Warning, math time. To get Tompkins' unweighted number, we simply add up the similarity value of each book and then divide by the number of books. So that's 1 plus 1 plus 1 plus 0.3, all divided by 4. That gives you an average similarity of any book to the same book in the other text. But if you wanted to consider the whole text, you can't use that number. You need to multiply each number by the length of each book, then divide by the total length. For this, we're using word count to keep it relatively simple. That means multiplying 1 by 33,002, 1 by 32,046, 1 by 30,147, and 0.3 by 855. Then dividing it by 33,002 plus 32,046 plus 30,147 plus 855. To reduce that down, you have to divide 95,451.5 by 96,050. You can see why this creates problems for the 85% number either way.
A huge difference from what scientists have been claiming for years.
Yep, turns out when you do the math wrong, you get big differences from what people who got the math right get.
And an 85% similarity really isn't surprising. Chimps and humans, and even dogs, cats, horses, and more, share some basic genetic similarities that specify proteins that help metabolize foods, grow hair, breathe air, and drink water. So of course, much of the genetic material would have to be the same since it specifies similar biochemical processes.
Okay, well, let's examine this for a bit. Chimps are morphologically and behaviorally generally more like the other apes than they are like humans. Based on this common lifestyle, common design hypothesis, you think that while humans might cluster with the great apes, that all other great apes would be more similar genetically to each other than any are to humans. But that's not true. The chimpanzee and bonobo are much closer to humans than they are to gorillas, and gorillas, humans, chimps, and bonobos are all closer to each other than they are to the orangutan. So here's a pic of a chimp, a human, a gorilla, and an orangutan. Would this hypothesis really predict that the orangutan is the odd man out? Really? No, of course it wouldn't. And it gets more extreme. Here's a goose barnacle, a shrimp, a silverfish, and a tiny mysticocarid. Except for the barnacle, everything here is a clearly segmented motile creature which has one clear head, a thorax, and an abdomen, and the barnacle is at most cryptically segmented, sessile, with no abdomen, and barely a head. And really, the barnacle, being at most cryptically segmented, sessile, and with no abdomen and barely a head, should be the odd man out of this group. Instead, It's the mysticocarid. A barnacle is much more genetically similar to a silverfish and a shrimp than to a mysticocarid. These animals are very different with very different lifestyles, so this hypothesis of similar lifestyle leading to genetic similarity falls apart when you actually examine it. That's what we call falsification.
But chimps don't have the intellectual abilities of humans to create or communicate, or the creative abilities in their hands. For example, they cannot build hospitals, operate on one another, play baseball, or write poetry.
Yes, chimps aren't humans, so they don't do human things. And humans can't do many of the things chimps can do. For example, chimps are much better at short-term memory, they can brachiate far better, and are much stronger than humans. Each and every single species on the face of the planet is unique, but so is every individual in that species. That doesn't make them not related, it makes them distinct.
So it makes sense that when new research compared individual pieces of chimp DNA piece by piece with similar places on human chromosomes, they were about 15% different.
Except no, they're like 2 to 4% difference when you do the math correctly, depending on your exact technique for counting differences.
That means at least 450 million DNA letter differences separate the two species.
No, it's about half of that, and since each species has its own history, really each lineage needs only about a quarter of those changes to get to where we are today. But hey, at least we're in the right order of magnitude, I think, but then there's no maximum number of genetic differences that two organisms that are related can have.
Far too many precise and vital bits of information for any source other than genius intelligence to generate.
Well, that was asserted without any evidence or argument, so I'm just going to say no.
Chimps and humans are very different, not only in their anatomy and behavioral traits, but also in their DNA.
Fun fact, very doesn't actually mean anything in particular. It's just a weasel word here.
When the DNA sequences of humans and chimps are compared, it becomes clear that they were each engineered uniquely and separately by an omnipotent genius creator.
Again, not even argued for. Next up, we have cell origins. Let's take a look.
What's one of the biggest problems for evolution? The origin of the first cell.
Is that a problem for evolution? You see, it's an open question, but questions aren't problems. Let's take a look at the difference with a different historical theory, the luminiferous ether theory. Before Einstein solved the problem of the speed and propagation of light, the dominant theory was that there was a substance called the luminiferous ether, which was the medium to light, like air is the medium to sound. Now, this substance, which was thought of as material, couldn't be stationary compared to Earth at all times, because Earth orbits the Sun. This means that if you measure the speed of light in different directions at different times of year, you should be able to get at least small differences in the speed of light depending on direction, essentially detecting the so-called etheric wind going past the Earth. Now, at this point, how fast is the etheric wind is a question for the ether theory of light. But when the Michelson-Morley experiment failed to find any etheric wind, it became a problem. There can't be no etheric wind at all if the ether theory is true. The lack of the wind falsifies the main aspects of ether theory. So you see, a thing a theory can't currently explain well is a question. If evolution has no explanation for how the first cell came about, that's not a strike against evolution. Now, if we learned that how the cell came about with high confidence and it contradicted how evolution works, then it would be a problem. Science is okay with not knowing things. That's what research is for. For some reason, though, creationists like to pretend that any area in science that has open questions, which, by the way, is all of them, can just be dismissed on the grounds that they don't know everything.
Though the cell is the basic building block for life, its design is actually very complex. We could compare the functions of a cell to those of computers, building projects, and even whole cities.
Loosely, yeah, but that doesn't really tell us anything about their origin, since we know that those things are designed, built, and used by humans, but we also know that that's not true for cells, and we can't put God in a test tube, so we can't simply assume that design analogies hold true in biology.
Coordinated machines within the cell access vital information in the cell's DNA. Certain protein machines transfer that information at just the right times and amounts to RNA molecules. And others use some of that information as blueprints to build more proteins. So it takes a protein along with DNA and RNA to make a protein.
This is simply incorrect. RNA can catalyze protein formation all on its own, provided amino acids, and both RNA and amino acids can form abiotically. This isn't a chicken and the egg situation. We know that of these, RNA probably came first, then proteins, then DNA. This part isn't even really an open question, never mind a problem.
Could random evolutionary events explain the origin of this three-way interdependent process?
No, but evolution isn't just random. It involves random events, in the form of mutations and drift. But selection is not random. So did cells form randomly? No, they did not, and evolution doesn't suggest that they did.
Some believe that the first cell arose spontaneously from a warm pond of chemicals.
I would love to see a citation for who thinks that the first cellular life just popped fully formed out of the primordial ooze. I bet it's basically no one in the field of origin of life research. Which, by the way, is a distinct field from evolutionary biology. You see, it appears that we're currently conflating abiogenesis and the formation of the first cell. The first life probably wasn't cellular, and since evolutionary biology requires populations of biological organisms to even exist, the formation of such things is fundamentally not a part of evolutionary biology. But then creationists like to just conflate any part of science they don't like with evolution.
But attempts to produce simple biomolecules from supposed early Earth environmental soups have produced nothing even close to the array of complicated chemical machinery that functioning cells use.
As we should expect. No model of abiogenesis involves a modern cell popping out of any prebiotically plausible chemical system. Even the simplest cell today is the product of billions of years of evolution, starting before there were even cells in the first place. This is just a misunderstanding on top of a straw man. It's really bad.
These experiments keep running into a stubborn fact. Natural processes tend to destroy the hardware of life rather than craft it.
Not really. Amino acids, RNA, sugars, and lipids all form naturally under prebiotically relevant conditions, not just in labs, but even in space. There's nothing magical about organic chemistry.
What about the software, the programming inside cells?
RNA came before DNA, and it only later started to build DNA as a more stable method of storing genetic material. But random RNA strings can undergo natural selection on their own, as well as reproduce on their own, and form proteins all on their own. Now, RNA probably wasn't the first kind of genetic material that ever existed, although what was is indeed an open question. But even starting with completely random genetic sequences will result in some differential reproductive ability, and boom, selection is off to the races. We don't need to introduce a programmer to explain this.
Evolutionary scientists shifted their attention to DNA, codes that tell cell parts what to do and when and where to do it. But biological codes are more complicated than computer codes. The true origin of life must account for the complex information programmed into the cell that directs protein machinery to do things like make more proteins.
No, it doesn't, because that's a job for selection, which is already known to explain such things in modern organisms, where we can see genes in real time arise out of what is essentially noise, as well as the modification of existing genes, sometimes after duplication, leaving two distinct genes with a common genetic ancestor.
So we can be certain that just like the most complex computers had designers, the cell also had a designer, the divine engineer who created all things.
Non sequitur. Your facts are uncoordinated.
Well, our third clip is the fossil record.
Do fossils really show that creatures gradually evolved from simple to complex?
Yes. Well, okay, our fourth clip is junk DNA, which I'm sure won't be dumb in any way. Not at all. Oh. You want me to cover the fossils, don't you? You're probably even hoping for a transitional species slideshow. Okay, I guess I'll do it then. First, while on the whole life has gone from simpler in the beginning to more complex now, that is not a necessary trend. Evolution can also work to simplify things. Most famously, parasites are often radically simpler than their non-parasite relatives. Perhaps the most extreme in this are the myxozoans, which are parasitic jellyfish that are so radically simple they only have a handful of cells, and in some lineages have even lost their mitochondria.
The late fossil expert and evolution defender Stephen Gould recognized they didn't.
That's just a lie, and I'm sick of giving all these professional creationists the benefit of the doubt on this. If you're going to cite someone, you need to actually know what they said and why they said it, and not misrepresent it. If you don't, and you just grab a quotation parasitically from another bottom-feeding creationist, you're lying. Period. In no way did Stephen Jay Gould think that the fossil record did not show evolution. The man, when asked about creationists' use of his quotations, said this: "Since we propose punctuated equilibria to explain trends, it is infuriating to be quoted again and again by creationists, whether through design or stupidity, I do not know, as admitting the fossil record includes no transitional forms. Transitional forms are generally lacking at the species level, but they are abundant between larger groups. Let's take a look at some of them.
He said the fossil record shows two clear features. One, sudden appearance, and two, stasis.
Yes, he did, specifically at the species level. But he also knew that transitional forms above that level were in fact all over the place.
When a new creature is discovered in the fossil record, it shows up suddenly and fully formed, with no gradual forms that led up to it.
I guess it's that time again, and so soon after the last time.
That's sudden appearance. Then its basic form stays the same. It doesn't evolve. And that's stasis.
Look, I'm not going to do it again for a third time, but yeah, it still applies. This is just, there are no transitional forms, the long version. But everyone knows that yes, there obviously are. It's like when flat earthers deny that the southern celestial pole exists. You can just look at it, but they don't care. It shows their absurd beliefs for what they are. Obvious absurdity, so they just hand-wave dismiss it.
All kinds of fossilized creatures show sudden appearance and stasis, including creatures like crinoids.
Oh hey, look, it's Tenoimbricata, a transitional echinoderm leading towards crinoids.
Mosasaurs.
Oh hey, look, it's Aegialosaurus, a transitional lizard leading to Mosasaurus.
Beardworms.
You know how they use that photo from modern beardworms? That's because we don't have any definitive fossils of them because being deep-sea organisms, they're extremely unlikely to show up in the fossil record. And so there's no way to show stasis through geological time because we don't know what they looked like any earlier than today. Weird choice of organism.
Horseshoe crabs.
Oh hey look, it's Lunataspis, a transitional xiphosaurian leading to modern limbulid horseshoe crabs.
Lobsters.
Creationists love to pick lobsters because despite people thinking of them as some special group, they're actually pretty primitive decapods. Lobsters are living transitional forms between shrimp-like organisms and crab-like ones. So any fossil intermediary is just going to be a robust shrimp, more or less. You can then just say, nope, it's a lobster, and move on.
And trilobites.
Trilobites have some of the most detailed evidence of evolution between species and families of any group in the Paleozoic. Claiming that they are an example of stasis is ludicrous. They can manage it by pretending that trilobite is some small group like lobster, a single family. But instead, it's actually a whole class. Grouping them together is like looking at humans, tigers, platypus, and kangaroos and saying, gee, mammals sure haven't changed much.
Rather than gradual evolution, the fossil record shows each animal form suddenly appears.
Hey, look at this picture of phyllid evolution with the geological extent of known taxa in dark and the inferred phylogenetic relationships in light color. But that's just like what we were shown and told indicates no evolution. But, creationists accept the evolution of these animals from a series of common ancestors in a branching hierarchy. So why is it that this fossil assemblage is fine as an evolutionary tree, but others aren't? Could it be, actually, that creationists don't have a standard to judge by, and all their ideas about kinds are just post hoc attempts to desperately salvage a view of the history of the world that hasn't been tenable for about 2 centuries? Maybe.
Trilobites have perfectly formed body parts
I don't even know what that means. What would them being imperfect look like?
right from the start.
From the start of what? The trilobite fossil record? The earliest trilobites are certainly not as heavily ornamented as later ones, so are they imperfect? What about Spergina, a potential proto-trilobite? Is it perfectly formed? Who knows? They're just weasel words that haven't been defined.
They show up suddenly with no ancestors
No certain ancestors, but we have potential ancestors.
and have some of the most complex eyes that ever existed on Earth.
I mean, they're compound eyes that uniquely use calcite for lenses. I'm not sure what about that makes them anomalously complex or something that couldn't evolve.
Does that sound like slow, gradual evolution?
Definitive trilobites fossils don't show up at the beginning of the Cambrian. It takes until about stage 3 for the first ones to show up in the fossil record. So that's 30 million years from the beginning of the Cambrian to our earliest known true trilobites. That sure does sound like gradual evolution.
They first occur in Cambrian rocks,
Yeah, stage 3 Cambrian rocks. Let's not forget that.
which often represent the first deposits from Noah's flood.
No, there was no flood. The flood, as believed by creationists, is physically impossible in so many ways that I can't even count them. I've covered this many times, as has friend of the channel Gutsick Gibbon. Go watch our back catalogs. I'm not going in depth during this section.
But even when found high above Cambrian rocks, trilobites still look like trilobites, as though they were created to reproduce more of their own kind.
Weird. Normally we'd hear that kind is about the family level, but Trilobita is a class. In fact, here's a slideshow of trilobites. Do those all look the same? They don't to me. In fact, that looks like a lot of evolution.
In The Origin of Species, Charles Darwin hoped future fossil finds would show examples of in-between creatures demonstrating evolution. So far, it's just more stasis.
Screw it. Here it is a third time, but faster.
Creatures appear suddenly, fully formed, and they keep the same form. And that's what we'd expect to find. God's fully formed creation we read about in the book of Genesis.
Odd about all those transitional species, then. Our fourth clip is about junk DNA.
Darwinists assume creatures evolved through random mutations.
Starting off strong with the word Darwinists. But again, mutations are stochastic, so is drift. But selection is not. And it is pretty darn important, especially since if you're going to focus on Darwin, all you really get is selection, given that he lived before mutation was understood, and before the words mutation and gene were even invented. That is not an assumption, it's an empirical fact. Humans have pseudogenes. Perhaps most famously, the human gene that would prevent scurvy is broken, as evidenced by, the fact that scurvy is a thing. But that's not the only gene that's broken, and there are whole regions of the genome that are physically incapable of being of any use to the organism. They are full of stopped codons, useless repeats, and degraded viral genomes, etc.
This leads to another assumption that human DNA must be full of useless sequences. They call it junk DNA.
Incorrect. Junk DNA is not simply DNA of unknown function that is assumed to not have function until demonstrated otherwise, nor is a full genome sequence necessary to estimate the functional fraction. Just plain population size combined with population growth rate of necessity introduces range of junk DNA mathematically required to be in that situation. Further, the existence of junk DNA makes predictions. One important one is that if you measure diversity of regions of a genome compared to the distance from known selected regions, that is, regions known to be functional because they're under selective constraint, you will see diversity jump up exponentially on average with distance until it reaches a maximum diversity based on effective population size. This is observed. Junk DNA is not a supposition. It has been independently demonstrated to definitely be there in multiple ways. There is a link in the description to a longer discussion of this by evolutionary biologist Zach B. Hancock here on YouTube. Zach has some great genetics and evolutionary biology content and is severely undersubscribed. He's also one of the science friends, so give his channel a look, and if you like it, subscribe.
In 2001, the Human Genome Project was published. This massive project took years, but eventually scientists sequenced the entire human genome. Secular scientists recognized about 5% of its purpose, but they couldn't figure out the other 95%. They quickly assumed it had no purpose. They labeled these unknown regions junk DNA and claimed them as evidence for evolution.
Nope, they didn't. They labeled them as unknown and had independent evidence of junk DNA.
More responsible scientists caution that this assumption came out way too soon.
It can't be a premature conclusion if it wasn't a conclusion.
We should find out if it has a function before declaring that it doesn't. The secular project ENCODE was launched to do just that. And in 2012, its scores of researchers announced that at least 80% of the human genome is functional.
You think they'll point out that in 2014, ENCODE walked that back? No, probably not. And why did they do that? Well, because they had a bonkers definition of function. They use what can be called the causal definition of function. This states that if x causes y, then y is the function of x, and x has function because it causes y. But let's see how that works in other parts of life. Most people know that the function of food in your fridge is to be eaten by you or your family. But let's say you buy some strawberries and put them in your fridge, and then you don't eat them in time, so they grow mold. Well, you won't be eating them, but hold up, are they functionless? Well, they're causing mold growth, so under the causal definition, the purpose of strawberries is actually to grow mold, so you shouldn't throw them out. They're functional food after all. I think you can see why this is, silly. So let's give some examples of what they consider function that are unreasonable. First, we have being targeted for methylation. Methylation causes parts of the genome to coil up tight and makes transcription virtually impossible. Methylated regions of the genome largely cannot be expressed, and it's hard to see how a part of a genome being locked away from any further biochemical activity makes that part functional. Another example is spurious transcription. A region that has a start codon followed by a few more codons and then a stop codon is called an open reading frame. But let's say you have a start codon, a few dozen other codons, and then a stop codon. That's probably too small to do anything but waste resources building a structure that can't meaningfully interact with the cell, because it's just a few amino acids in a string. It's essentially scrap. It's only useful to be broken down to how it started, but since both building and breaking it down costs energy on net, it would be better if it hadn't been made at all. It's hard to call this functional, but for ENCODE, that's biochemical activity caused by a region of the genome, so it's functional. You can see why basically every geneticist who looked at ENCODE's 2012 number of 80% functionality rejected it, and why in 2014 they greatly reduced this estimate. Turns out, the critics were right.
And this percentage will likely go up.
It had dramatically gone down by the time this video was produced. It had already dramatically gone down by the time this video was produced.
Other evolutionists objected, saying that these results must be wrong since they contradict Darwinism.
They don't contradict Darwinism because Darwinism doesn't have anything at all to say about genomes, mutation, functional fraction, etc. And further, it's not just that they contradict the predictions of evolution, they also contradict independent measurements of functionality as well as the basic math of population genetics. And lo and behold, when critics looked, they found that the reason they contradicted both theory and previous results is that ENCODE was using a pretty silly definition of function.
No way should DNA have that much function. It should be full of useless junk.
Which it demonstrably is.
But the ENCODE results don't lie.
Yeah, those 2014 results that walked back the 2012 numbers sure don't. Odd we still haven't heard about that. It's almost like creationists just ignore findings they don't like.
Most of our genetics are not junk. They were coded with top-notch performance, just like we'd expect from an all-powerful designer.
Well, given that we know for a fact that the majority of the human genome is junk, if their prediction of design is little to no junk, I guess that's design falsified. Our fifth so-called fact is called wonderfully made.
Nothing is more precious than a newborn baby, and perhaps no greater wonder exists than a human life growing within another human life. A baby thrives in a total water world for 9 months, which is utterly impossible for any person to live in immediately after they take their first breath.
Something to do with not having an umbilical cord, I think.
A baby in the womb has blood vessels with a different arrangement and structure than an adult's. This helps him get enough oxygen to grow.
Yep, that was the placenta and umbilical cord that they showed. This isn't really news, but I'm waiting for a point, and I'm really hoping that the claim isn't just abortion bad. That's an opinion.
The baby also produces hormones that cause the mother's body to meet the baby's specific needs. The placenta, a temporary organ, takes nutrients from the mom's bloodstream in such a way that the baby's needs are met first. And in the last weeks of pregnancy, the baby releases even more hormones to tell the mom's body he is ready to be born.
Well, if you have things are cool or complicated, therefore God, you can mark that on your bingo sheets, I guess.
Mother and baby are deeply connected, yet two very separate beings in an all-or-nothing reproductive system.
What does it even mean to be all or nothing and step-by-step in this context? As opposed to what? Embryology without steps? Gestation that doesn't finish but produces a viable organism anyway?
A step-by-step evolutionary process couldn't lead to something so complex.
Oh, it's just irreducible complexity. So the claim that something cannot evolve can be defeated by any plausible evolutionary history, whether that history is actually what happened or not. So can we come up with a plausible way for placental reproduction to evolve? I think so. So let's go through it. The most basic form of sexual reproduction that we'll start with is isogamy, in which an organism releases gametes that are undifferentiated, and any two gametes can merge to produce a zygote. This is a better strategy than just cloning because it allows for crossing over and greater diversity, both of which increase average fitness for the next generation. It has some waste though. For one thing, self-crossing is extremely likely, and self-crossing is still better than cloning, but not nearly as good as outcrossing with other individuals who are not closely related. So differentiating gametes into distinct classes, perhaps by size, can reduce this problem. It also then allows one class, the small one, to become motile, which enhances the chance of fertilization while still limiting self-crossing. We now have male and female gametes. Next, it may become advantageous for some species to differentiate into male and female individuals, because there are advantages to each. Reproduction for females is more costly since the larger the gamete, the more the cost of producing it from a biological standpoint, but females are more likely to reproduce successfully on average than males. Males have a lower cost of reproduction, but are more likely to fail, so it's a give and take either way. But also, having distinct sexes completely eliminates the problem of self-crossing. Now, the energy needed to keep the embryo going until it can feed itself has to be contained in the egg cell, so there is a selective pressure to make eggs bigger and bigger until we get large macroscopic eggs with yolks or yolk analogs. Remember, an ostrich egg is a single cell, and it's enormous. So now, we are at the point that most fish and amphibians are at. Now, the closer to the egg laying the fertilization occurs, the more likely both the male and female are to have their gametes successfully result in zygotes. So there is pressure to move from simple broadcast spawning to external fertilization with the male and female closely paired. Well, this also creates pressure for some extension of the reproductive system to help direct gametes, and of course, the closer the association is between the male and female, the higher the fertilization rate is. So now that organ directing sperm can become an intromittent organ for internal fertilization. Now, of course, we're at the point where we are almost at amniotic reproduction. We just need a coating around the egg to protect from water loss, and we need it because the animals we're talking about were moving onto land as parareptiles. The amniotic sac provides extra water, and the shell is just a collection of biominerals and proteins that covers it. So, we're at the point of how lizards and monotremes reproduce. Now, in mammals, parental care starts to increase because it allows for fewer offspring, but having a higher rate of survival for each one. One way to help with this is to limit the time offspring spend as eggs external to the mother's body. That way she can protect them longer. This means that eggs will be held internally for longer and longer, which is what we can see in the platypus, which while it lays eggs, only does so towards the end of gestation, and the eggs hatch very quickly after being laid, but quite a while after being formed. It's only a small step to go from this to just letting the eggs hatch internally and the young be born live. This occurs in many species of lizards, although we're focused on mammals. At this stage, the eggshell is a waste of resources, so there's pressure to stop making them, and we get shellless but amniotic internal eggs that hatch inside the mother. Unfortunately for the mother, though, many fertilized eggs will not make it to full term. In humans, about 3/4 of all fertilizations of an egg by sperm will not make it to term. So any resources that go into egg yolks and the like for such eggs are a waste. Simultaneously, any egg that can attach itself to the uterine wall and get some extra resources that way is more likely to survive and have a higher birth weight, which is an obvious advantage. So we have two big selection pressures encouraging embryo implantation on the uterine wall and getting nutrients that way. And with mammals that make milk, it's also possible for these babies to be born fairly underdeveloped, because they can nurse. So we're basically at the proto-marsupial level of reproduction, where there is no placenta, but there is still implantation, and the young are born very underdeveloped, but can use milk to continue their development, all with extensive parental care. And every step has been beneficial so far. But even more beneficial is the option to stay internally in the uterus longer through longer implantation. But this creates a problem. The mother's reproductive system is more and more likely to destroy the embryo the longer it is implanted. That means that suppression of the immune system during pregnancy, as well as mechanisms to increase the efficiency of resource transfer to the embryo, are both selected for. In fact, the placenta in mammals is actually tissue of the embryo and not the mother, and is derived from the tissues that would create a yolk. While I skipped by it a bit in marsupials, they do have a short-lived placenta, but that doesn't change much, and either way, we're at human-style placental development, with every stage being selected for positively, and nothing that wasn't stepwise. So yes, the placenta can indeed evolve. Did it follow this exact sequence of events in reality? I don't know, but it doesn't matter. The argument that it can't evolve is defeated.
God put these special features in place so that we could not only survive, but thrive. The psalmist declares, "For you formed my inward parts. You covered me in my mother's womb. I will praise you, for I am fearfully and wonderfully made."
In a way that's completely consistent with evolution. Well, the sixth so-called fact is evolving bacteria.
Doctors tell us that keeping our hands clean is essential. Why? Because bacteria is everywhere.
Bacteria are everywhere. The word is the plural form of bacterium.
On things you touch, in the air you breathe, in the food you eat, and even living inside you. Some of these microbes are good for us, like those that help us digest food or fight off infections. But with all the bad bacteria out there, scientists are working hard to develop new antibiotics to combat them. Even then, bacteria often mutate when they reproduce, making some of them resistant to medicine and dangerous to our health.
Gee, a mutation that increases fitness in a particular environment that then comes to dominate the population? That sounds a lot like evolution, gotta say. Is this just them saying, well, bacteria evolve, but they're the only ones? Are they going to say that doesn't count because reasons? Let's find out.
Is this process evolution in action, like some claim? Hardly.
Yes, evolution is at a minimum a change in allele frequency in a population across generations, and that's what happened.
That's because researchers have discovered that bacteria are, well, still bacteria.
Yes, that's because you can't just escape your ancestry. Mammals will always produce mammals, bacteria, vertebrates, humans, etc. Evolution produces branching. New branches can emerge, but they always do so within the context of previous branches. Objecting that bacteria remain bacteria is to fundamentally fail to understand the most basic aspects of evolution.
For instance, in a 20-year study of E. coli, which can cause food poisoning, scientists looked at 40,000 generations of the bacteria and confirmed that the E. coli did not evolve into anything other than E. coli.
This is a reference to the Lenski long-term E. coli experiment, and it's actually quite wrong. You see, bacteria, because they generally reproduce asexually, can't really be classified using the biological species definition. Instead, they are assigned to species by physicochemical properties, in a form of the morphospecies concept. One of the really important diagnostic characteristics of E. coli is that they cannot metabolize citrate in the presence of oxygen. But in this experiment, the line now called CIT+, has evolved that ability, meaning that they no longer meet the criteria for being E. coli. Now, they haven't been given a new specific designation to my knowledge, but that's in part because that's not really the point of the experiment, but still, they are no longer classically the same bacteria.
A similar study used fruit flies, which are made of many more cells than bacteria. After 600 generations, about 12,000 human years, they were still just fruit flies.
Again, monophyly. You can't just stop being a fruit fly if your ancestors were fruit flies. You can become new types of fruit fly, even ones that barely resemble what we think of as fruit flies. But you'll always be a fruit fly.
And for scientists who carefully examine the Bible, these results make a lot of sense. That's because God created each creature with the ability to reproduce only after its kind.
Say it with me. What's a kind? It's whatever the creationist needs it to be. For humans, it's a single solitary species. For most animals, it's a family, whatever that is. For bacteria, it's a whole domain with more diversity than you can find in all of Animalia put together. It's just a weasel word to let creationists accept the evolution they can't deny, while denying the evolution they can't accept.
So, bacteria will never be anything other than bacteria, which means you still need to wash your hands.
Yep, as required by evolution. Well, we're on to the 7th alleged fact. This time, it's the Cambrian explosion.
It's the Cambrian explosion.
If evolution were true, the fossil record should show one type of animal life in the lowest layers, then 2, then 4, and so on.
Well, that's cool, because in the lowest layers that have any evidence of animals, we have only scant evidence for the presence of sponges by chemical signature. Then as we get to the Ediacaran, we see more, but still very few, and then in the Cambrian, that increase gets turned up to 11, which is, what we'd expect. But that graph was labeled phyla, which is not how this works. While there are some relatively hard definitions of species, all taxa are fundamentally arbitrary, even species. Phyla aren't some special fundamental aspect of biology. They're just a group of large, but again arbitrarily named, taxa. So what should we expect if we could look at every organism that ever lived and get a full view of the real phylogeny of life? Is that early on, at about the time of the beginning of the Cambrian, or at the end of the Ediacaran, that each speciation we would see would be perfectly ordinary at the time, but that the lineages that result from them, that still have descendants, would end up being what today we would call different phyla. But the Cambrian is an adaptive radiation that resulted in many speciation events, many of which were almost certainly not well preserved, which means that we should see a large number of phyla coming about more or less at the same time, which is what we do see, although it's more or less at the same time than more.
But Darwin himself had a difficult time explaining what we now call the Cambrian explosion of life.
Darwin is still not the Pope of Science. He's not a prophet of evolution. He's still not Rasul al-Ilm. It doesn't matter what he could explain. Current evolutionary biology has no trouble explaining the Cambrian, and while, like in all things, there are open questions, as we've already discussed, there are no problems.
Cambrian rock layers contain nearly all the basic animal forms, including echinoderms, arthropods, and even chordates.
But not Birozoa, Rotifera, or unambiguous onychophorans. So, that's a bit of a misstatement. But again, we expect this early in the history of life. With an adaptive radiation, we should see numerous phyla appear in the Cambrian. But they still don't all appear at the exact same time. Arkarua adami is a possible echinoderm from before the Cambrian, back in the Ediacaran. Rusophycus appears about 537 million years ago and is the oldest known arthropod, so it's 3 million years after the start of the Cambrian. The earliest chordate we know of is Bacchaia gracillans, from 505 million years ago. The thing is that all these phyla do appear in the Cambrian, but implying that this means simultaneously ignores the fact that in reality there are tens of millions of years between the divergence of these phyla from their stem groups. It's simply not true that the earliest members of most phyla appear simultaneously. Instead, it is actually true that they appear over time through the Cambrian mostly, but some phyla are even present earlier, such as Porifera.
With no trace of transitional fossils in lower Precambrian rocks.
That's not even true for their on-screen examples, as we've already gone over in this video.
And each fossil exhibits amazing complexity.
Complexity is not a distinguishing factor between special creation and evolution. Both predict things will be complex, although it's actually the happenstance and contingent nature of evolution that predicts higher degrees of complexity rather than design, because designers tend to strive for the least complexity needed, and organisms in many cases are more complex than strictly needed.
For example, Cambrian arthropods had advanced compound eyes, like we see today in dragonflies and bees.
Cool, but Precambrian probable stem arthropods did not. So again, that's what we'd expect to see.
The eyes were tuned to the way light travels through water, allowing the creature to see straight.
I'm sorry, is this argument that an animal adapting to its environment is evidence that a process that causes organisms to adapt to their environment wasn't operating? That seems more than a little silly.
This required each eye to have over 3,000 interconnected lenses.
I guess we can fill in big number, therefore God on the bingo cards, even though 3,000 isn't actually that big a number, and a stock photo of an exaggeratedly surprised woman isn't going to change that.
Compare that to only one lens in each human eye.
And humans have many more photoreceptive cells per lens. Vertebrate and arthropod eyes are complex in different ways. It's like they weren't even designed by the same designer. Maybe different designs, different designers, right?
Cambrian rocks also contain fossil fish that look like today's lanceolids and hagfish.
The earliest chordates are a lot like the most basal chordates. Isn't really the anti-evolution flex that ICR seems to think it is.
The fish had perfectly crafted skeletal supports inside their bodies.
Kind of. Neither the earliest chordates, lampreys, or lancelets have bony skeletons. Instead, they have a scaffold of cartilage.
An opposite arrangement from Cambrian arthropods like trilobites.
Oh, once again we see different design. But for some reason, that doesn't imply different designers. Weird that. Evolution predicts that organisms that diverge early on will end up with significant differences.
The Cambrian layers show an amazing amount of diversity and complexity.
Amazing doesn't mean anything useful. If the claim is that it's too much diversity, you actually have to do some work to show that that's the case. And so far, everyone who's tried has failed to do so.
Evolutionists claim it happened in a mere 5 to 10 million years.
No, more like 40 million to 60 million, but hey, who's counting, right?
The equivalent to a blink of an eye in so-called geologic time.
Kind of, but a short time for a rock is a very long time for a group of organisms.
Sudden appearance? Instant complexity? That doesn't sound much like evolution.
Or much like the actual fossil record.
Instead, the fossil record shows fully formed creature groups right from the start.
Fun how they glossed over all the stem groups like radiodonts or lobopods that no longer exist and show that in fact modern phyla arose within wider evolutionary groups in a radiative adaptation from which only a few groups persist to the present day. Which is, exactly what evolution predicts. But anyway, our eighth, fact is apparently the Big Bang, which is a fact, but I'm guessing they're going to say it's not.
How did the universe begin?
Our whole universe was in a hot, dense state that nearly 14 billion years ago expansion started.
Some people say that it came into existence billions of years ago in a massive explosion.
Yeah, by some people, he means essentially everyone who makes it their life's work to study cosmogony. AKA all the people who know what the hell they're talking about.
But this Big Bang has some big problems.
Who wants to bet that by problem he means open questions? Although there actually are some problems in cosmogony to do with the expansion rate of the universe and how old exactly that makes the universe.
First, the antimatter problem.
Yeah, I'd call this a problem. Current models predict equal amounts of matter and antimatter.
Whenever scientists convert energy into matter, they get equal amounts of matter and antimatter. Antimatter particles have the same mass but opposite charge.
And spin, but whatever.
Supposedly, the Big Bang converted energy into equal amounts of matter and antimatter, but the universe shows no sign of this expected antimatter.
Yep, and that's why current models must be flawed in some way. But what about this means that the universe was never in a hot, dense state that then expanded into the modern, relatively cool and sparse state? Oh, right. It's nothing. This is like saying you can't tell someone how to cook a steak unless you know exactly how bovine embryology works. No, the fact that you don't have a full picture of how a steer got onto your dinner plate doesn't mean that what you do know is wrong. Maybe I'm hungry.
Second is the horizon problem. The Big Bang model says that the universe started out very small and rapidly expanded. This would leave huge hot and cold spots, since even a 13 billion year old universe is still too young for temperatures to level out. But the actual universe shows temperatures remarkably the same in all directions, no hot or cold spots.
Yep, that's why the original version of the Big Bang was modified to include cosmic inflation, which results in a much more isotropic universe. That's the thing science is supposed to do, modify its ideas when presented with new data. Now, when was the last time creationism did that? Oh, right, basically never, at least in any significant way.
Mature galaxies pose a third problem. Light from the farthest galaxies we see supposedly took billions of years to get to Earth. So those distant galaxies should still appear undeveloped and chaotic, as they would have after the Big Bang.
No, in fact, the earliest galaxies discovered are indeed primitive and non-arranged the way modern galaxies are, including in the formation of galactic clusters. This is right on NASA's website.
But distant galaxies look fully mature, not haphazard like the Big Bang predicted.
Except, when they don't, which is the case for the earliest galaxies, like I already said.
Even the very distant regions of the universe reveal galaxies of magnificent beauty and complexity.
Okay.
In addition to these scientific problems, the Big Bang contradicts the Bible.
Cool. I don't care. If you have empirical evidence, then you have a seat at the scientific table. The Bible isn't that. Our ninth and antepenultimate fact is our young universe, which sounds a lot like the one we just covered, but let's see what's up.
Just how old is the universe?
About 14 billion years old if we are to round to the nearest billion.
Well, it doesn't have an expiration tag, but we can make some estimates by looking at things in our universe, like the moon.
How the f*** are you going to look at the moon to see how old the universe is? That's like looking at someone's fingernail clippings to see how old they are. Fun fact, the moon isn't thought by any scientist to be primordial, so no matter how young it is, that means nothing for the age of the universe. But let's see what they have for us anyway.
As the moon orbits the Earth, its gravity pulls on the oceans, causing tides. And as the tides pull on the moon, they cause it to move about 1.5 inches away from the Earth each year. That means that 6,000 years ago, the moon would have been about 800 feet closer to the Earth. Not a problem. But if the Earth and Moon were over 4 billion years old, then they would have been touching, and that wouldn't have been good.
No creationist has ever actually managed to give the math on this claim, and when I run it every way I can think of, I've failed to get that result. Since this claim is universally made without evidence, I'm fine with dismissing it by simply saying, math or get the f*** out.
So the moon shows that the universe is several thousand years old.
Nope, even if it were a claim that had any evidence, which again, it doesn't, the Moon-Earth system isn't the universe, and it doesn't have to be as old as the universe as a whole. If you find a juvenile barnacle on the shell of a big sea turtle, does that mean the turtle is no longer than the baby barnacle? No, of course not. That's stupid. That's what's happening here, except at least juvenile barnacles are real, and this problem isn't.
That makes sense when we also look at comets.
Has anyone ever said that comets popped right out of the Big Bang? No, no one has said that. But let's hear some creationists pretend that short-period comets show the universe is old because they just hand-wave dismiss that there are long-period comets that are the source of new short-period comets, while pretending that young comets mean young universe for literally no reason.
They lose large amounts of material each time they pass around the sun, so they wouldn't even be around after billions of years.
Oh, they're not even going to acknowledge that there are in fact explanations? Neat. That's about as low effort as it is possible to get.
And the inner regions of spiral galaxies rotate pretty fast. If the galaxies, and the universe altogether, were billions of years old, they'd be too twisted up to look like they do now.
Nope, because spiral arms are actually standing pressure waves that are not formed from a set grouping of stars. But stars enter and leave these areas, and they rotate as a whole arm at a rate different to that of any of the stars in them. They are created by objects like dwarf galaxies. They are not vortices or something like that.
So everywhere we look in the universe, we see evidence that shows that what the Bible says about its age is consistent.
Odd that there were no examples given. Our penultimate topic is the Goldilocks planet.
Scientists have been looking for the Goldilocks planet for years. The name obviously comes from the popular Goldilocks fairy tale. Now, the Goldilocks idea, much like the fairy tale, is all about finding a planet that's just right for life. But finding the right planet is harder than you might think, especially when you think about how special the Earth is. Earth is the only known planet in our universe that can support life. Earth has water, animals, and people, all necessary to support life.
Wait, animals and people are necessary to support life? That is just absurd. But that's what they said. I really wish they'd make a point. We're already like halfway through this topic.
Earth is just the right distance from the sun. If we were any closer, it'd be too hot. And if we were any further, we'd be freezing.
Actually, no. Estimates for the habitable zone of the sun range from about half the distance of Earth to the sun to about 3 times the distance. It's not just that any body within that range would be habitable. It also needs the right gravity, magnetic field, albedo, and atmosphere. But when it comes to just the distance from the sun, Earth is towards the inside of the habitable zone and could be almost as close as Venus or as far out as the asteroid belt and still be at least potentially habitable.
The Earth also has just enough gravity to support an atmosphere, and yet not so much as to crush us.
Given that people can do just fine in hyperbaric chambers, I'm going to go ahead and say that the range is also quite large and that Earth could be quite a bit bigger without having a crushing atmosphere. Now, you might say that gravity would be too high, and yeah, it might be for humans as they exist on Earth, but they are adapted to Earth gravity. Life on a high G world would be adapted for that gravity. so something could live there, even some intelligent observer.
Take any other planet. Mars has a very thin, unbreathable atmosphere of carbon dioxide. Venus has a thick, crushing atmosphere with clouds of sulfuric acid, and Mercury is just way too hot.
That uninhabitable planets exist doesn't mean that habitable planets can't. I'm not sure what the point of this is.
Not too hot and not too cold, not too heavy or too light, but just right for life, as created by a loving God.
Nothing in this even hinted at Earth being indicative of a miracle. It didn't even talk about the most unusual aspect of Earth that enables life. A large moon. But hey, that's their argument to make, not mine. The final topic of this video is 3-in-1, whatever that means.
For centuries, philosophers and theologians have tried to explain the Trinity, the reality that God is Father, Son, and Holy Spirit. He is three persons, but one God.
I'm not here for theological debates. This section is entirely an analogy for the Trinity, using various aspects of the universe. Sure, whatever, I don't care. We're skipping it. Next time we'll start off with the 7-day week, so that'll be fun. Well, if you enjoyed the video, hit like and tell me in the comments. If you didn't, feel free to hit dislike and tell me what the problem was, again, in the comments. Either way, please remember to subscribe and hit that bell icon so you're always notified when I have more content. I'm the Dapper Dinosaur. Thanks for watching, but before you go, I just want to say a special thank you to my channel members and my patrons, especially those pledging $20 or above. Bob Knob, Tapioca Weasel, Klaus Jepsen, Bent Hovind, Kevin Brostek Van Manen, Landon Knoll, Mabbity Babbity, Monkey They Them, San, Denny5252, Eleron Teller, Phil Cavara, Ian Chen, Mrs. Spexinder, Sphincter of Doom, The Venerable Bede, and Scriptic Mystic. As you probably know, YouTube is a very volatile platform, and from month to month my income on the channel can swing wildly. But the people you see on screen are directly supporting so that I don't have to worry too much about that, and the channel can keep going as it is, and perhaps even improving. If you'd like to join this team, there are links below to join the channel or in the description to join the Patreon. The Patreon starts as low as $1 a month, and the channel memberships start as low as $1.99 a month. On Patreon, you can even get a discount for pledging annually. If you do decide to pledge, you'll get access to an exclusive Discord server just for channel supporters, as well as early access to all of my pre-recorded videos, often up to 2 months in advance. Higher tiers will unlock even better perks. Now, if the annual or monthly pledge isn't right for you, but you still want to support the channel, there is a merch store down in the description, as well as an Amazon wishlist, just for me. And if financial support isn't something you can or want to do, then if you still want to help out the channel, please just like and share these videos and make sure you comment on them. It really helps the channel out. Thanks again for watching.